Dear students, in last video we have discussed about biotechnological application in the field of agriculture. We have discussed BT cotton and pest resistant plant tobacco. Okay, so today in this class we shall study biotechnological application in the field of medicine. Okay. Biotechnological applications are many in the field of medicine. Mainly, the technique involved in the production or synthesis of medicine is recombinant DNA technology. In previous chapter, we have studied about the process of recombinant DNA technology. Using the same technology or technique, we produce medicine. Using recombinant DNA technology or technique, we are able to produce safe and more effective therapeutic drugs on large scale to improve human health. So, what is the meaning of this sentence? Using recombinant DNA technology, now we are able to produce many drugs. These drugs are more safe drugs. These drugs are more effective. It means these therapeutics or the drugs are very specific drugs. Very specific means particular drugs. And these drugs do not produce or do not induce any unwanted immunological responses. Unwanted immunological responses means what we say in common word, side effects. There is no unwanted effects by taking these drugs. Because the drugs are very specific drugs. So these drugs improve the health of human beings. Right now, at present, nearly 30 recombinant therapeutic are approved for human use. Through genetic engineering, we are able to produce 30 recombinant drugs or therapeutics, which are even approved for human use all over the world. Among 30 drugs, 12 drugs are presently using in India. The number is specific, very important. ನಾವೆಲ್ಲ ಈಗ ನೋಡ್ತಾ ಇದ್ದೀವಿ COVID-19ಗೆ almost all countries in the world, they are doing research in order to find out a vaccine for COVID-19. Still, all these vaccines are under clinical trial test. ನೀವೀಗ ನೋಡ್ತಾ ಇದ್ದೀರಾ, ಯಾವ ದೇಶನೂ ಇನ್ನು ಫೈನಲ್ ಆಗಿ ನಾವು ವ್ಯಾಕ್ಸಿನ್ ತಯಾರು ಮಾಡಿದ್ವಿ ಅಂತ ಹೇಳ್ತಾ ಇಲ್ಲ. ಮಾಡ್ತಾ ಇದ್ದೀವಿ, ಟೆಸ್ಟ್ ಮಾಡ್ತಾ ಇದ್ದೀವಿ, ಕ್ಲಿನಿಕಲ್ ಟ್ರಯಲ್ ಟೆಸ್ಟ್ ನಡೀತಾ ಇದೆ ಅಂತ ಹೇಳ್ತಾ ಇದೆ. ಯು ನೋ ದಿ ಪ್ರೊಸೆಸ್ ಆಫ್ ಡೌನ್ ಸ್ಟ್ರೀಮ್ ಪ್ರೊಸೆಸಿಂಗ್. ಯಾವುದೇ ಒಂದು ಡ್ರಗ್ ಮಾಡಿದಾಗ ಆ ಡ್ರಗ್ಸು ಕ್ಲಿನಿಕಲ್ ಟ್ರಯಲ್ ಟೆಸ್ಟನ್ನು ಪಾಸ್ ಆಗಬೇಕು. ದೆನ್ ಓನ್ಲಿ ಇಟ್ ಈಸ್ ಯೂಸ್ ಇನ್ ಹ್ಯೂಮನ್ ಬೀಯಿಂಗ್ಸ್. ಓಕೆ, ನೆಕ್ಸ್ಟ್ now we shall see about insulin. I am going to give little bit extra information. Okay. Insulin is a human protein. It is nothing but an hormone. This is synthesized. Insulin is synthesized in pancreas, that is, in the cells of islets of Langerhans. So insulin hormone is produced or synthesized in the cells of islets of Langerhans in pancreas. What is the role of this insulin in human body? This insulin maintains the glucose level in the blood. Glucose level in the blood means the sugar level in the blood. Deficiency of this hormone or the insulin cause what is known as hyperglycemia. Hyper means more. See, sugar level increases in the blood. So if the insulin is less, it causes hyperglycemia. Hyperglycemia means the sugar level is increased in the blood. This condition is called diabetes mellitus. Common ಆಗಿ we call diabetes ಅಂತ ಕರಿತೀವಿ ನಾವು. Because of deficiency of insulin, the sugar level is more in the blood, we call blood sugar. Similarly, the excess sugar is also found or secreted in the urine. This condition is called glycosuria. So this is the reason why doctors test blood and urine. ಡಯಾಬಿಟಿಕ್ ಇರ್ತಕ್ಕಂಥ ಅವರಿಗೆ ಬ್ಲಡ್ಡನ್ನು ಮತ್ತು ಯೂರಿನ್ನು ಟೆಸ್ಟ್ ಮಾಡಿಸ್ತಾರೆ. ಕಾರಣ ಏನಂದರೆ ಬ್ಲಡ್ ಶುಗರ್ ಜಾಸ್ತಿ ಇದೆಯೋ ಯೂರಿನ್ನಲ್ಲಿ ಶುಗರ್ ಜಾಸ್ತಿ ಇದೆಯೋ ಅಂತ ನೋಡ್ಕೊಳ್ಳಲಿಕ್ಕೆ 2 ಟೆಸ್ಟನ್ನು ಮಾಡಿಸ್ತಾರೆ. ಓಕೆ, ನೆಕ್ಸ್ಟ್ earlier means before the invention of the insulin or the discovery of insulin. Insulin is used for diabetics or extracted from pancreas of slaughtered cow or pigs. How the diabetic patients obtain this insulin earlier? They obtain from pancreas of slaughtered cow or the pigs. It caused allergy or other type of reactions in some patients. Why caused allergy means there is little change in the structure of insulin. Change in the structure means the number of amino acid. So this is the reason why the patient got allergy or the reactions. Okay, it's also important for the examination. Next, we shall see the structure of human insulin. ಇನ್ಸುಲಿನ್ ಹೇಗಿದೆ ಅಂತ ನಾವು ನೋಡೋಣ ಈಗ. ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ಹ್ಯೂಮನ್ ಇನ್ಸುಲಿನ್. See the diagram. There is a diagram in the textbook. See the diagram. Diagram is also important. Insulin consists of 2 short polypeptide chains. Alpha chain, which you can say A chain, with 21 amino acids, and beta chain with 30 amino acids. See the diagram. The matured insulin consists of 2 polypeptide chains. A chain we call alpha chain with 21 amino acids, and B chain or beta chain with 30 amino acids. These two chains are linked together by disulfide bond. See the diagram now. In mammals, including humans, it is synthesized as proinsulin. This proinsulin should be processed for maturation and function. The proinsulin or the hormone contain extra chain. See the diagram. C chain. C peptide chain. When it is removed, then it becomes functional insulin. Very important point. The proinsulin consists of A chain, B chain, and also C chain. If the C chain is removed, then it becomes functional insulin. Okay. So this is the insulin that controls sugar level in the blood. Okay. That's all about the structure of human insulin. Now we shall see very important point for the examination. Explain the synthesis of insulin through RDNA technology or genetic engineering. ನೀವೆಲ್ಲರೂ ನೋಡಿ ಇನ್ಸುಲಿನ್ ಪ್ಯಾಕೆಟ್ ಮೇಲೆ ಥ್ರೂ RDNA ಟೆಕ್ನಾಲಜಿ ಅಥವಾ RDNA ಅಂತ ಬರೆದಿರುತ್ತೆ. ಅಂದರೆ RDNA ಟೆಕ್ನಾಲಜಿ ಇದ್ದ ಡೆವಲಪ್ ಮಾಡಿರ್ತಕ್ಕಂಥ ಇನ್ಸುಲಿನ್ ಅಂತ. ಸೊ ದಿಸ್ ಇನ್ಸುಲಿನ್ ಈಸ್ very good for human health because it has no side effects. Okay. Now, synthesis of insulin through genetic engineering. Very important for 3 or 5 marks. It was in the year 1983. For the first time, Eli Lilly, an American company, prepared 2 DNA sequences of A and B chain of human insulin. It means the company synthesized artificial DNA, that is DNA of A chain and DNA of B chain. Now earlier ಆಗಿ ಹೇಳಿದ್ವಿ ನಾವು synthesis of DNA in the lab is also a technique of biotechnology anta. You can see now. This company synthesized 2 DNA molecules, aDNA and bDNA molecule separately. So this gene is inserted into the plasmid of E. coli. How? Using restriction endonuclease enzyme. You know, restriction endonuclease enzymes. If plasmid is treated, it will cut the plasmid and produce sticky ends. Then the gene, a gene, is introduced into the plasmid. B gene or the DNA also introduced into the plasmid separately. Thus obtained a recombinant DNA. That is, DNA contained human insulin gene. This technique, that's why called recombinant DNA technique. Because of the gene, human gene in the plasmid of E. coli cells produce a chain. That is, the cell, E. coli cell having a gene produce a chain. The E. coli cell with B gene produce B chain of insulin separately with beta galactosidase protein. So this beta galactosidase is produced because of the gene. Because of the gene, it is produced. So that beta galactosidase gene, that is removed. How it is removed? It is treated with cyanogen bromide. Then we will get A chain and B chain. Okay. Thus obtained proinsulin contain C chain, which is removed with the treatment of trypsin. Remember, these 2 points are the need points. Beta galactosidase protein is removed with the help of cyanogen bromide, and the C chain is removed with the help of trypsin enzyme. Then both the chains A and B chains are extracted and then linked together by disulfide bond. Now the insulin is as good as human insulin. We have already studied the structure of human insulin. The human insulin consists of A chain and B chain. These two are linked by disulfide bonds. Thus, human insulin is produced. This is the insulin. Now, all over the world, the diabetic patients, they are taking. So this insulin controls the blood sugar level. So this is the insulin which is synthesized through rDNA technology. So thus produced insulin is genetically synthesized human insulin, and it is called humulin. Sometimes very important in the NEET examination. Humulin andre ಏನು? Nothing but human insulin. So this is how the human insulin or insulin is synthesized through recombinant DNA technology or rDNA technology or using genetic engineering. Okay. ಈ ಥರ ಇನ್ಸುಲಿನನ್ನು ತಯಾರು ಮಾಡ್ತಾರೆ ಜೆನೆಟಿಕ್ ಇಂಜಿನಿಯರಿಂಗ್ನಿಂದ ಅಥವಾ ಬಯೋಟೆಕ್ನಾಲಜಿ ಇದ್ದ. ಈ ಇನ್ಸುಲಿನ್ನ ಆಲ್ ಓವರ್ ದಿ ವರ್ಲ್ಡು ಉಪಯೋಗಿಸ್ತಾ ಇರೋದು ಈಗ ರೈಟ್ ನಾವು. ಓಕೆ, ರೆಫರ್ ದಿ ಡಯಾಗ್ರಾಮ್ ದ ಟೆಕ್ಸ್ಟ್ ಬುಕ್. The textbook diagram is very important even for the main examination and also for CET. CET-ನಲ್ಲಿ ಆ ಡಯಾಗ್ರಾಮ್ ಕೊಟ್ಟಿರ್ತಾನೆ, ನೀಟಲ್ಲಿ ಇರೋದಿಲ್ಲ, CET-ನಲ್ಲಿ ಇರುತ್ತೆ. ಸೊ ದಟ್ಸ್ ಆಲ್ ಅಬೌಟ್ ಸಿಂಥಸಿಸ್ ಆಫ್ ಹ್ಯೂಮನ್ ಇನ್ಸುಲಿನ್. ಇದು ಒಂದು ಬಯೋಟೆಕ್ನಾಲಜಿಯ ಅಥವಾ ಜೆನೆಟಿಕ್ ಇಂಜಿನಿಯರಿಂಗ್ನ ಅಪ್ಲಿಕೇಶನ್ನು ಇನ್ ದ ಇಂಪ್ರೂವ್ಮೆಂಟ್ ಆಫ್ ಹ್ಯೂಮನ್ ಹೆಲ್ತ್. ಓಕೆ, ದಟ್ಸ್ ಆಲ್ ಅಬೌಟ್ ಸಿಂಥಸಿಸ್ ಆಫ್ ಇನ್ಸುಲಿನ್. ನೆಕ್ಸ್ಟ್ ನಾವು ನೋಡ್ತಕ್ಕಂಥದ್ದು ಜೀನ್ ಥೆರಪಿ. ಓಕೆ, ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸಲ್ಲಿ ಜೀನ್ ಥೆರಪಿನ ನೋಡೋಣ. ಥ್ಯಾಂಕ್ ಯು.